Hello everyone. Today we will discuss about the plant tissue culture. What is plant tissue culture? Plant tissue culture is a technique of growing plant cells, tissues, organs, seeds, or other plant parts in a sterile environment on a nutrient medium. Now we will know about the plant cells are totipotent. That means uh, we will now discuss about the totipotency. Totipotency is the ability of a cell or tissue or organ to grow and develop into a fully differentiated organism. Totipotency, also called totipotentiality, refers to the ability of many cells to retain the full genetic potential characteristics of the mature organisms from which any of the cells were obtained, and thus the enable the cell to regenerate the organism's tissues and organs under favorable conditions. The possibility of regenerating an entire plant from a single or few non-zygotic cells was proposed by Gottlieb Heberland in 1902. Heberland is now popularly called the father of tissue culture. How? Adult plant cells are totipotents, meaning they have the ability to give rise to a fully differentiated plant. Because of this, it is possible to collect cells from a mature plant and use those cells to produce clones of that plant. In the picture, you can see that the root of a carrot plant, and then we collect some root cells in the growth medium, then the single cell after cell division in culture we have many cells, and from this culture a young plant can be produced and ultimately which resulted in the adult plant. Why do plant tissue culture? A single explant can be multiplied into several thousand plants in less than a year. This allows fast commercial propagation of new cultivars. Taking an explant does not usually destroy the mother plant, so rare and endangered plants can be cloned safely. Once established, a plant tissue culture line can give a continuous supply of young plants throughout the year. Virus-free explants can be cultivated to provide virus-free plants. Plant tissue banks can be frozen, then regenerated through tissue culture. Plant culture in approved media are easier to export than soil-grown plants as they are pathogen-free and taking up little space. Tissue culture allows fast selection for crop improvement because explants are chosen from superior plants, then cloned. Tissue culture clones are true to type as compared with seedlings, which show greater variability. Now, why we should do tissue culture? The production of clones of the plants that produce particularly good flowers, fruits, or have other desirable traits. To quickly produce mature plant, the production of plants in the absence of seeds or necessary pollinators to produce seeds. The regeneration of whole plants from the plant cells that have been genetically modified. The production of plants in sterile containers reduces disease transmission, allows production of plants from seeds that otherwise have very low chances of germinating and growing, such as orchids, to clean particular plants of viral and other infections, and to quickly multiply these plants as cleaned stock for horticulture and agriculture. Now, the plant tissue culture basics. Modern plant tissue culture is performed under aseptic conditions. Living plant materials from the environment are naturally contaminated on their surface with microorganisms, so surface sterilization of starting material in chemical solution. Explants are then usually placed on the surface of a solid culture medium but are sometimes placed directly into a liquid medium when cell suspension cultures are desired. Culture media are generally composed of inorganic salts plus a few organic nutrients, vitamins, and plants' growth hormones. Plant tissue culture basics. It has as a culture as cultures grow, pieces are typically sliced off and transferred to new media to allow for growth or to alter the morphology of the culture. Then, what's the history of the plant tissue culture? In 1838, Swan and Sladen put forward a theory which states that cells are totipotent and in principle are capable of regenerating into a complete plant. Their theory was the foundation of the plant cell and tissue culture. 1902, Gottlieb Heberlandet conducted the first but unsuccessful attempt of tissue culture using monocotyledons. He first gave the concept of totipotency in tissue culture. He is popularly called as the father of plant tissue culture. 1926, F.W. Went demonstrated that there were growth substances in colleptiles from oats. 1934, what generated continuously growing culture of meristematic cells of tomato on medium containing salts, yeast extract, sucrose, and three vitamins. This established the importance of additives in tissue culture. 1939, successful continuously growing cultures of carrot and tobacco. 1948, formation of adventitious shoots and roots in tobacco. 1952, virus-free dahlia through meristem culture. 1952, first successful micrograft. 1953, Miller and Cook discovered kinetin, a cytokinin that plays an active role in organogenesis. 1953, haploid callus of ginkgo biloba produced from pollen. 1954, first plant grown from a single cell. 1957, root or shoot formation in culture depends on auxin-cytokinin ratio. 1958, regeneration of somatic embryos in vitro from citrus. 1958, pro-embryo formation in callus clumps and cell suspension of carrot. 1960, enzymatic degradation of cell wall for protoplast formation. 1960, vegetative propagation of orchids by meristem culture. 1962, development of the Murashige and Skoog medium. 1965, induction of flowering in tobacco tissue in vitro. 1966, Stewart demonstrated totipotency by regenerating carrot plants from single cells of tomato. In the 60s and 70s, Murashige cloned plants in vitro, raised haploid plants from the pollen grains in many plant species, first achievement of protoplast fusion. In the 70s and 80s, beginning of genetic engineering. Now, the plant tissue culture applications. The commercial production of the plants used as potting, landscape, and florist subjects. To conserve rare or endangered plant species. To screen cells rather than plant for advantageous characters, such that herbicide resistance, tolerance. Large scale growth of plant cells in liquid culture in bioreactors for the production of valuable compounds like plant-derived secondary metabolites and recombinant proteins used as biopharmaceuticals to cross distantly related species by protoplast fusion and regeneration of the noble hybrid, to produce clean plant material from stock infected by viruses or other pathogens. Production of identical sterile hybrid species can be obtained. And that's it. Thank you all.